Welcome to my world. Another day, another time, another period for knowledge acquisition. Knowledge is power, health is wealth. Today's topic has to do with exploiting telomere for longevity. To exploit something means you have a full knowledge of it, and then you now want to apply it to make it possible. Telomeres generally are on every chromosome, and they are involved in the divisions of cells. The growth of every cells or development comes about by these divisions, and for every division there is a shortening of these telomeres. If we can prevent the shortening and elongate it, it is possible to extend, to elongate the lifespan. So it's in a nutshell, it will affect longevity. So how do we, the researchers or medical laboratory scientists, get involved in these researches to help humanity elongate the lifespan? That is what I shall be looking on as usual. If you like my presentation, press the like button, subscribe, and be sure to join me in the slide section. The outline: objective, introduction, research findings, there's so many, lab diagnosis, and finally recommendations. The objective: to exploit some research findings that associate a balanced diet with telomeres length in order to standardize a normal value for every age group in a given society for their longevity. The introduction: every living cell consists of chromosomes, which are the fingerprints that make for our physicality as individuals. Every cell has a cycle of growth and division to multiply and occupy more space that is manifested in their physicality. This multiplication is called meiosis in somatic cells, diploid cells, that is having the two, and meiosis in germ cell, haploid, that is instead of 46, 23, 23. Telomeres is the protective cap of every chromosome, and its length is shortened with every cell division, thereby gradually exposing the chromosome to mutation. The protective nature of these telomeres is gradually reduced, and finally the cell dies off, and it's part of what aggregate to contribute to the death of even the person. In 1984, Elizabeth Blackburn discovered the molecular structure of telomeres, and together with Carroll, both discovered the enzyme telomerase that lengthens telomeres. Its DNA is specific for the telomerase. According to Kimber, leukocyte telomerase length is influenced by a complex interplay of lifestyle and heritable factors. The heritability of healthy hair is estimated to be up to 83%, although this varies significantly and depends on family history. You can understand some family have old age as something inherited. Telomere length can be measured using southern blood telomerase restriction fragment, flow cytometry with fluorescence in situ hybridization, and quantitative PCR assay. As demonstrated by Leonard Effe a half-century ago, human cells have a limited replicative lifespan, with older cells reaching this limit sooner than younger cells. We could also compare this early limit to the contact phenomenon, where cells, when they reach the end of whatever and they make contact with the next cell or the marker, they stop dividing and stop multiplying. This Elfrig's limit of cellular lifespan is directly related to the number of unique DNA repeats found at the end of the genetic material bearing chromosome telomeres, which safeguard the ends of chromosome from unwanted and unwarranted DNA rearrangement that destabilize the genome. Each time the cells divide, the telomeric DNA shrink and will eventually fail to secure the chromosome ends. So you can see the telomeres in the diagram below. You can see how it is the reverse transcriptase producing the increasing the number of the tandem repeat, the DNA tandem repeats as once it's there or stimulated or however it is triggered, it's now start multiplying that tandem repeat of that specific telomeres, so it lengthens it back. But when it is a cancerous growth, then you can understand why there is no contact phenomenon. They continue to multiply, not stopping. The research findings. 1. The human holoenzyme telomerase is a ribonucleoprotein composed by a catalytic unit and an RNA component, which act as a template for the addition of a short repetitive sequence in the 3-end direction of the telomeric DNA and species-specific accessory protein. 2. The telomerase is composed of two main subunits, the telomerase RNA component named TAR and the telomerase reverse transcriptase protein, and a variable number of accessory factors. In most normal cells, telomerase activity is low, which leads to gradual telomeres shortening. This process is called replicative senescence, whereas in most cancer cells, telomerase activity is high, which allows cells to divide without limits. Telomeres length and telomerase activity are important for cancer initiation and survival. Researchers are developing drugs that inhibit telomerase as a potential cancer treatment. And data from epidemiological studies and clinical trials have demonstrated that the increased intake of micronutrients such as vitamin D, C, E, and A, and dietary fibers in the context of a balanced diet, just as I mentioned in the objective of this project. E.g., example, the Mediterranean diet is associated with longer telomeres, as reviewed by Gallic et al. Yeah, there are so many research findings, and each cell, some will tell you so much, like this, that each cell division leads to shorter telomeres due to the inability of DNA polymerase to fully replicate the end part of the lagging strand of DNA, described as the end replication problem. Telomere attrition and cellular senescence are identified as major contributors to the physiological process of aging. This is another finding by somebody great. Furthermore, increased aging and age-related diseases have been shown to be associated with disruption of telomeres homeostasis, which has been mainly attributed to increased level of oxidative stress and inflammation. So we could change up to the research on the association of telomeres shortening with age has revealed that the inhibition of telomeres attrition via telomeres gene therapy in mice result in delayed physical aging. Furthermore, critically short telomeres have been shown to be associated with higher mortality rates and shorter lifespans. That's another finding by great men, scientists. And we therefore can come to the conclusion that telomeres activity can be positively modified by natural compounds such as astragalus metrobenous plant extract, cycloastaginel, and curcumin CR, and vitamins, among others, which enable telomerase activation. And that's a finding reported as by another great scholar. So all this helps to pinpoint that if we can look at the way of checking our telomeres and see how we can increase it, we can also help in increasing our lifespan. That is where the recommendations now come in. Lab diagnosis. The laboratory is a veritable place for doing great researches, and to really check for telomere length, it can be measured using southern blot telomere restriction fragments, flow cytometry with fluorescence in situ hybridization, and quantitative PCR assay. There are so many tests you can do, and it has also been found that telomere length at 25D is a very strong predictor of realized lifespan. Those individuals living longest had relatively long telomeres at all points at which they were measured. So a comparative analysis could help us find the range and match it with age. So people, just like the body mass index does, people can aim for that target. Finally, the recommendations. From some of our research findings so far, we have seen that a balanced diet shows a strong relationship with telomeres length, and it is now left for researchers to prove which of the content of a balanced diet really specifically elongates telomeres. With that, if that could be found, then that will be the area of concentration for people to maintain their telomeres length. The negative effect of stress and inflammation on telomeres length confirms their role in the aging process, more or less saying that stress is not good, and once we have inflammation in any injury, we must make effort to really correct and become normal again. We have learned that with telomeres length intact, the reactive oxygen stress will be unable to damage them. The DNA causing many of the chronic diseases of old age. Once the telomerase that covers and protects the chromosome is still intact, the DNA would not be exposed to damages. That is exactly, and the chronic diseases of old age would still be a far cry. It will not be there. It will just be more or less like an acute experience that the children and the young adults usually have. More work needs to be done to avail us the opportunity of standardizing the telomerase length at a particular age to encourage laboratory testing for telomerase, like it's done for blood hemoglobin level or body mass index. I learned that some international developed countries are already on this, that they could really test for the length of the telomerase and also guess the age from telomerase. So with all that in place already, it means that we are getting gradually to having lifespan elongated for humanity. If we can maintain our telomerase length, then we can become ageless and safe from all the detrimental effect of an unbalanced diet. So we are associating the balanced diet with telomerase, and once we know the specific substance that we could take as supplement to the diet, we could always use that and maintain our telomerase length. This does not rule out dying from various accidents or mishap, which is not in consideration of this study. Please, if you like the work done, press the subscribe more, so that more work can easily be done. The encouragement needed will be there. And press also the like button, and you can watch so many. It is now about 168 videos within 2 years.